Welcome to the Electra-Med Edge, where we explore the science, technology, and innovations advancing human performance and patient care through precision measurement. I'm Andy, Clinical Sales Manager at Electra-Med, and I'm excited to have you join us as we launch this new series. Electra-Med is proud to be a service-disabled, veteran-owned small business founded in 1975 as a biomedical service organization. Nearly 5 decades later, our mission remains the same: to deliver not only cutting-edge equipment and supplies, but also the support of the sale, meaning we're here for our customers long after installation and training. Today, Electra-Med provides full-service solutions across neurology, cardiology, physical medicine and rehabilitation, sleep diagnostics, respiratory care, and U.S. government contracts, serving clinicians, researchers, academics, and performance professionals nationwide. Through Electra-Med Edge, we'll bring you conversations with experts, insights into emerging technologies, and real-world applications from across these fields, all aimed at advancing human performance and clinical precision. Let's get started. Hello, good day to you all out there in podcast land. My name is Andy. I'm the Clinical Sales Manager with Electra-Med Corporation based in Flint, Michigan. We are the premier sales service provider of Lotus cycloergometry. We also deal with respiratory, sleep diagnostics, physical medicine, batteries, and a whole other aspect of equipment sales and service. Today, we have Dr. Robert Musci, Assistant Professor of Health and Human Sciences at Loyola Marymount University in Los Angeles, California. He is in the Health and Human Sciences Department. Currently, Dr. Musci's research focuses on applied geoscience and exercise lab, interested in translating and understanding mechanisms to target aging in humans, not so that we live longer, but to improve the quality of life of individuals. Other aspects of research is measuring mitochondrial bioenergetic capacity using high-resolution respiratory and measuring mitochondrial DNA turnover using stable isotopes. Looking forward to jumping into this episode. Dr. Musci, thank you for joining us.
Thanks for having me. Appreciate it.
Absolutely. So, Dr. Musci, maybe for all of our listeners and everything, you can just give a little bit more of an explanation of, some of your work, what your primary role and things like that are at Loyola Marymount University.
Sure, yeah. So Loyola Marymount University is a primarily undergraduate institution, which means we take teaching very seriously, and we use research as a means to help facilitate that teaching. So I'm really split 50-50 when it comes to teaching and research. And so as a teacher, I focus predominantly on exercise physiology, not necessarily to prepare people to become exercise physiologists in the future, but to really help our future healthcare workers understand the integrated physiological nature of the human body in health and disease. And we use exercise as that platform to understand that. And taking that in the lab, we also see exercise as this really phenomenal way to improve what we call the health span, time living free of disease and being able to move around. And so we look at how exercise influences our body cellularly, molecularly, and how that affects overall functional outcomes. So we focus mostly on mitochondria, central to metabolism for the cells. It's linked to many health outcomes in humans, and we're looking at how certain interventions, exercise being one of them, can target both mitochondria, improve protein quality within the skeletal muscle, and then improve human function across the lifespan.
That's awesome. No, I think that, and this is really a foresight into kind of the future of how we can utilize at the mitochondrial level of how things are. Yeah, mitochondria is the powerhouse of the cell, right? So looking at how those integrated bioenergetics all integrate within one another. So that's awesome. I'm really looking forward to this. So maybe we can expand a little bit more on the applied physiology and your testing teachings. How does that fit into your curriculum, in your research? How does that blend together?
Yeah, certainly. So again, when we train our students, we want students to kind of have this holistic view of what human health looks like. And so for us in the lab, while we might be working with cell culture or rodent tissues or rodent preclinical models, when we translate these interventions or we're investigating processes in humans, we got to understand how these molecular outcomes impact physical function. So we use metabolic carts. We use bike ergometers. We use treadmills, of course. We'll measure glucose homeostasis and glucose metabolism using continuous glucose monitors. But then we'll also do DEXA scans to measure muscle mass or estimate muscle mass so that we can have this idea of function per unit of tissue, which is really important. And so it's really designed to try to link what students might physically see in a human, but it's someone's VO2 max or leg strength or power output, with some of the more molecular measures that they might seem a little bit more abstract and intangible, and try to bring the intangible to the visible, to the visible eye to them.
Sure, absolutely. No, it's interesting. And whether it be through mapping or through some other generation of that, that's an interesting way to translate that into it. So you mentioned using ergometers, and one thing I'd like to touch on. So currently you're using a Lodi Excalibur Sport. Maybe you can shed a little bit of light on how that functions and how that really brings your research a little bit farther into the future, maybe assists with your current agenda as well as maybe your future research agendas.
Yeah, certainly. So yeah, using a bike ergometer is really nice, and one, being able to conduct graded exercise testing. It's a lot. I would view it as a little bit safer in clinical populations. It's a little less intimidating than trying to increase the speed and grade on a treadmill for
Sure.
clinical populations as well. And so being able to use a device that interfaces with our software really makes the control of that ergometer much easier. It makes data collection much easier. It makes processing much cleaner, which is really nice from a research perspective. And while I'll pivot really quickly, because it is so much part of my job too, to the teaching aspect. And so in our exercise physiology lab, we have like the traditional Monarch bikes, and we have some without the light sensors. And so students in the very beginning of our anaerobic power lab have to manually calculate the distance traveled during a wind gate, and they have to measure the amount. They have to calculate the amount of force that was moved to get power output over that period of time. And so we take like sort of the basic physics and like the very simple approaches so that they get that experience, but then recognize that it's really important for them to have a clean experience and perhaps a better opportunity to process and understand the data. We like to move then to something that's a little bit more modern that can give us, really good data. That's not relying on someone trying to track someone's, foot as it goes around really, really fast.
Right.
That's been really nice too, to be able to show students where we have come from as a field, which is just over 100 years old, to where we are now. And it provides a historical perspective, but it also gives them a whole learning experience, so they don't just get a number and then they just repeat that number. They understand where the number comes from.
Absolutely. Well, it's funny, you mentioned VO2 max earlier, and my first thought was the original Douglas bag. Right. So
yeah, exactly.
Right. Hey, think of where we've come from that. And now the evolutions of our metabolic carts, they're portable, they're wearable. You can do them in field testing now. So you really see that those leaps and bounds within, like I said, that last 100 years or so. That's awesome. And I think you make a good point, because that allows students, I think, to start to develop their own research questions as we get into this, and maybe figure out like, oh, what else could I explore? Or what else are we interested in? Obviously working with experienced faculty like yourself, but do you happen to see, maybe because of the work you're doing and the research you're involved with, students are coming to you and looking to explore or interested in exploring something else, or a segue off of what you are currently doing?
Yeah, and that's sort of the really exciting thing about how we have structured this department and kind of structure our classes. Is, again, I want to keep on talking about exercise physiology lab because I'm pretty proud of that one.
Sure.
But at the very end of the semester, the students have a 4-week-long project where they have to come up with their own research questions. And so it's really fun to see as they learn how to use the equipment to come up with their own creative ideas, try to pull what's in pop culture into the lab, so to speak.
Yeah.
And 4 weeks is not very long to pursue a research question, but get that experience of science. What is really nice is we start transitioning towards more sophisticated equipment. So focusing on like with the PFM, we can measure right and left power. We measure cadence as well. And so one research group here is interested in ACL recovery after ACL reconstruction surgery.
Sure.
And they're interested now as to what, seeing whether or not the decrement in power output or this imbalance persists even months and perhaps even years after the surgery's taken place. And so that's one example of like how better equipment, more precise equipment, more sophisticated equipment can really unlock opportunities in the future to understand where the decrement is in someone's human health, where and how that predisposes them to further injury or future injury, and perhaps evaluate better interventions that can better address that deficiency.
Oh, absolutely. And I think this is where a really good integration between whether it be physical therapy, obviously having to go there for post ACL therapy, but then also integrating exercise physiology into this, and you're getting into that return to play a little bit, right? So greedily, you can look at it on the bike, like, hey, how is that quad to hamstring strength ratio, especially on the pedal force, as well as is there more force production left to right, and you can really start to lay into that. So that's really interesting. Is there a way to also, with your research in mitochondrial, is there any carryover or any interest in collaboration in that regard?
Yeah, certainly. So I'm collaborating with my colleague who's just right next door, Dr. On, who is interested in the rehabilitation trajectory, and
Okay.
in concert with an orthopedic surgeon in the area, we are interested in return to play because that is so critical for athletes.
Sure.
But again, also just for, I mean, we think about in the realm of exercise physiology, like athletes are really, really cool. They're
Yes.
a small subset of the population, but so much of what we can learn from athletics can be applied to, us normal people.
Sure.
So yeah, absolutely. Linking metabolic outcomes is a very keen interest of ours. And so one of the things we're also doing is piloting a non-invasive approach to evaluate muscle oxidative capacity
Okay.
using near-infrared spectroscopy. And so that would be an attempt to see if we can link metabolic deficiencies or discrepancies between right left, and also help guide return to play, and kind of understand the magnitude of detraining that occurs pre-post injury or pre-post recovery, because that's really important. One of the other projects I'm involved is very much addressed in the older adult population. One, can we predict risk of hospitalization based on metabolic outcomes, say
Oh, interesting.
mitochondrial respiratory capacity, or any name your biomarker of interest, whether or not that predicts hospitalization. But then importantly, does that predict recovery in the hospital or getting out of the hospital sooner, or does this person need extra care afterward? And so using non-invasive measures paired with precise measures is a really powerful way to better understand trajectories during injury or adaptations during training, and just getting a better sense of the physiology of humans.
Sure, absolutely. So, and let me ask you this, because that's an interesting, do you happen to see, or in your view, I mean, rephrase it that way, this is kind of a big gap, or there's still, again, we're always trying to further the science, but what I'm trying to get at is this one of those unanswered questions in exercise physiology so far of how do we get to these metrics, or how do we get to solving this question?
Yeah, so as a scientist, I think we're always trained to say, oh yeah, there's always a gap. And certainly there is. I think the challenge is trying to define what those gaps are. And I think in the field of wearables, I think they've advanced so far in the past 10 years where we think, oh yeah, we now have all these great capacities to measure all these sorts of different outcomes. And I'm excited by that, but I also still think we're still a far way away of understanding how we can use the measures we have now to better guide clinical intervention, or treatment. How do we use all the data that we're collecting to better inform our practice? And so, while I am excited for wearables, I also know that we still need very deliberate and well thought out experiments that are linked with not just the wearable signal, because I see that as I see wearables as a good signal, with some grounded physiology, both molecular and functional, and link that to a clinical outcome. And I think that's where I think the field really needs to focus and lean in is how do we take the tools that we have started to develop and move it to clinical improvement?
Well, that's a great question to try and figure out an answer, right? So we're getting there, right? But again, this is the fun part of science and how it's being impacted with, again, with research like yours at the mitochondrial level and figuring out what else needs to occur from it. So I do want to circle back a little bit. We touched on this off show, but coming back in, so we mentioned your work, but maybe you could again highlight a little bit of the integration and the experience you've maybe even all but brief with global proteomics as well and how that can impact your discoveries and future work.
I think my experience and my trajectory has really reflected the great aspect of science, and that is to say, we think we know something based on how we've measured this for decades, but as soon as you change the approach, you realize you don't know anything. And so, the question of protein turnover has been something that I've been interested in. It's what I was trained in during my graduate education, and it's something I'm still pursuing. So one of the biggest advances in the past 2 decades was the utilization of what are called stable isotopes. So in this case, deuterium oxide is just a heavy water, and we can use heavy water because it's safe for human consumption, and we can use that to measure protein synthesis, but also DNA synthesis and lipid synthesis over a long period of time. And my mentors, have spent a lot of work and effort. They've done a lot of work in this space, and so have others across the world. And they've refined these methods now to one, understand that the turnover of a protein is quite unique from all the other proteins. So when you talk about proteomics, we're talking about thousands of proteins. And in the past, we used to just measure the bulk, like just a mixture of these proteins. We had no idea as to how many proteins were in there, which proteins were in there, were the proteins that we were interested in, were they in there? And so now with the advent of what we call kinetic proteomics, the measuring of individual protein synthesis rates, we can look at the synthesis of a myofibrillar protein, of a specific myofibrillar protein versus a specific mitochondrial protein. And what's even more remarkable is that these proteins have different turnover rates. They're differentially affected by exercise. They're differentially affected by age and by disease. And so all of a sudden, you have this huge appreciation, say, when we say that with age, there's a loss of muscle mass. We call that sarcopenia.
Right.
The driving hypothesis has been, well, it's because protein synthesis slows down. It's we're not making as many proteins. But then the question must be asked, which proteins are slowing down? And so there's been a lot of preclinical work focused on showing this discrepancy. One, protein synthesis is not simply down. It's that some proteins are down and other proteins are up. We've only started measuring this in humans. So applied to mitochondria. Mitochondria are not these unique kidney beans. They're these crazy organelles that have weird projections, and they're kind of wrapped around in the muscle and all other sorts of different cells. And the proteins turn over at totally different rates. And so my lab is interested in how do mitochondria remodel themselves in response to stress, age or exercise, whatever you want to think of stress to be. And then link that to those functional outcomes.
Well, that's interesting. So I'm curious about this. I'm going to ask a little more of an in-depth question here. So when we talk about stress, right, are we talking an acute bout of whether, we'll just use a Wingate test, for example, right? That's a 30. Now, would we put grandma on a Wingate? Maybe not, right? But would we, if you're trying to induce a high intensity acute stress, i.e. a Wingate, was that what we're talking about, stress? Or could we also would it also be relative to, say, like an endurance stress as well? Maybe again, 10K or 15K, something like that.
Yeah. So when we talk about, we can talk about stress. Stress can be so many different things. And what's important to understand is that stress is often overlap. So there's plenty of research that shows that if you stress, a cell in one way, it has similar effects as like stressing a cell another way. So stress is a very broad topic. So then we talk about exercise, and it causes, increases heat, it decreases oxygen concentration a little bit, increases CO2, changes pH, changes, stresses energetically. And so when it comes to, say, bouts of exercise, I think there's a lot of excitement to try to better prescribe exercise intervention to elicit certain outcomes. I
Okay.
certainly see that in the realm of endurance athletes or athletes in general. And I think there's a shift now towards, especially with the exercises medicine movement with ACSM,
Yeah.
better being able to better prescribe kinds of exercise training or kinds of specific stresses to elicit better improvements, say on cardiovascular outcomes or metabolic outcomes to say prevent diabetes. And so I think there's movement and motion towards better understanding the stress responses to a single bout of exercise, to a series of exercises, or varying intensities. Um,
Gotcha.
we don't ask that question in our lab, but we are really interested in the trajectory of that response, right? So
Sure.
we don't get better from a bout of exercise instantaneously. It
Right.
takes a huge transcriptional response. We have to upregulate a bunch of genes. Those genes have to get synthesized into proteins, and those proteins have to get shuttled and moved over time. And that takes weeks and months. And we know adaptation takes a long time. But
Right.
we're interested in understanding the trajectory of that and then predicting based on what happens on bout one or bout two, what might happen on bout 15, a month or two later.
Sure, sure. No, this is very interesting. I'm very interested in this aspect. So I'm going to dive boil a little bit deeper here.
Yeah.
So you mentioned earlier like substrates and substrate utilization, whether we're talking about fats, a mix of carbohydrates and fats, or solely carbohydrates. Do you, in your experience, do you see someone who is better at substrate utilization, whether it be a power athlete versus maybe a general population, weekend warrior, quote unquote, versus, a trained endurance runner who is better at filtering and utilizing those specific substrates at those energy intensities?
It's not something that I generally look towards quite a lot in our
Okay.
lab. Certainly, I mean, right now we're working with a couple of sub-elite running groups,
Okay.
and it's always a fun contrast to see those who don't train all that often,
Hmm.
and contrast those to sub-elite athletes or elite athletes. And you see just substrate utilization to travel, say, 4 miles an hour or 10 miles an hour. And you can just see the vast difference in capacity to use fats versus carbohydrates in those very highly trained individuals compared to those who are not trained. And that's just, that's a consequence of just years long adaptations that, we see an increase in mitochondrial density with training that facilitates better fatty acid oxidation. We see improvements in running economy that just improves the efficiency of movement in these elite athletes. Um,
Sure. And again, this is forecasting into the future, right? But if we take
yeah.
that elite population as they are today, say they're in their late 20s, early 30s, do you think, with your experience thus far and what you've looked at in terms of protein turnover, do you think there's any, and correct me if I'm wrong, like any protective effect as they would get into older adulthood, things that, or like as they're setting kind of the foundation now versus somebody who maybe was sedentary in their mid 20s, 30s, now they've become more active in later adulthood?
Yeah, I mean, so I think there's been some papers that have highlighted and followed Olympians of the past and see how they age in the future. And I think it depends on the type of, excuse me, type of athlete. So
Okay.
there are some athletes that get super injured in their early 20s and they kind of have a really bad go of it as you get along. If you can maintain that health and maintain function and keep on training throughout your 20s and 30s, it seems to have a protective effect.
Hmm.
What I generally try to teach people and emphasize is that exercise at any age is always going to be very effective at protecting against most of your chronic diseases. So
Sure.
that's Alzheimer's disease, certain cancers, type 2 diabetes, cardiovascular disease, stroke. I mean, that the major ones that people are most concerned about.
Right.
There's a previous case study on an individual um, who lived in France. I believe he was 101 years of age
Oh, wow.
and
Okay.
still pretty active, but he went through a pretty dedicated training regimen for a year, and they measured his power output and his VO2 max pre and post this intervention. I think he rode something like 5,000 kilometers, so about
Oh, wow.
3,100 miles over the course of a year.
Sure.
So about 10 miles a day. Um, and they found that his VO2 max, even at the age of 101, still improved from exercise.
That
Still increased by about 10%. His power output, his maximal aerobic power output was about 25% greater, and in some cases actually higher than some of the students that I've measured in my exercise physiology lab, some 20-year-olds.
no
So
kidding.
what I tell people is age is, even though, yes, age is a huge factor in our health, there's a lot of variability in the aging trajectory of individuals. And the
Sure.
best way to bend that trajectory towards a positive outcome is to move more. And any movement is going to improve your stress resilience, is going to improve if people are interested in mitochondrial health, it improves your mitochondrial health. But more importantly, it just decreases your risk for all sorts of diseases and improves quality of life.
Sure. Absolutely. No, moving more is say at the end of the day, that's all you have to do, right? Whether it's a
Yeah.
simple couple 10-minute bouts spaced out, or ideally that would be the best, getting more bouts, even if it's the duration is less, but at that interval, but which is good. So no, this is great. And I really appreciate how you're thinking forward to the future, working with, and we're all going to age, right? There's no secret to stop time, but how you're trying to get into this. So, what I'd like to move in next is while we're speaking of the future, how do you see technology? We talked about wearables, but how do you see technology shaping how yourself and other educators prepare future clinicians and performance professionals?
Yeah, that's a really good question. I think what we've seen, I think you and I probably trained, kind of probably about the same age. So we're used to working with really clunky machinery,
Yes.
kind of the 1.0 versions of this of equipment integrated with computers. And I thought that was great back in the day,
Yeah.
but there were a lot of barriers to being able to easily analyze that data. And I think over the past decade, we've seen equipment shrinking. We've seen it
Mhm.
be a little bit more accessible for more labs. The software has improved. And so I think in many ways, we have this great opportunity of being able to collect more data. It's a matter of being able to process it accurately and interpreting it effectively. And so for us, not only are we worried about students being able to see what good data look like,
Mhm.
but also being able to analyze the data effectively and then interpret it again into an actionable outcome. I think
Absolutely.
what's really important, like you can't just throw a metabolic cart in a cardiology lab and say, "Hey, do a graded exercise test," and just read out the numbers and there you go, make that determination. The point of educating this next generation of our workforce is to make sure that they are empowered to understand what a good test looks like, be able to look at equipment and understand if it's working or not, if it's
Sure.
calibrated or not, if the numbers that you're seeing make sense, and then being able to confidently interpret and communicate those data to your patient or to your client. And so for the field, the challenge now is to effectively integrate technology into patient and client outcomes.
Sure. No, absolutely. I agree with you on that one. And quick side story here. You remember when you and I are probably the same time, but the old VO2 Max mouthpiece with the big headset thing?
Yeah,
Yeah.
absolutely. Yep.
And now they've got the nice silicone mask and it's a one
Hmm.
tube and you don't have to worry about anything.
Yep.
Yeah, interesting that no, it's great because you really do see how this technology is excuse me, shaping and allowing for these future professionals to really dig deeper into this and digging into it more and to say, okay, we know what is typical looking data and what it should look like. Therefore, we would be able to better recognize what is atypical or non-normal data and synthesizing that out and really having that ability for the data to tell the story at the end of the day, which is great. So, and we talk about equipment a lot. We talk about your research and everything. But another question I have is, how do you see industry and academia maybe collaborating a little bit more effectively to actually move the science forwards?
Yeah, I think that's a really good question. So really good question. I think I think the U.S. research infrastructure overall has a good model of public versus private support, and
Mhm.
I think sometimes we don't appreciate how much investment is made on either end.
Sure.
But when it comes to improving collaboration, I think it's just better communication as to what
Okay.
each field needs,
Sure.
and where each sector can assist in the driving of development. When I think about mechanistic research, I very much think about drug development.
Oh yeah.
You can come up with targets in a research academic lab quite easily. Maybe not quite easily, but like it can be done.
Mhm.
What's really difficult for a single lab to do is to run that into a clinical trial, right? And that's where like interfacing with industry is so critical and so important. The classic example right now is with GLP-1s, stemming from research in Gila monster venom. Like that was the initial discovery, and even before that, it was like I think fish that were supported some of that background research. So like the arc of like of research paying off into like real world intervention is very, very long. So the other thing I'd say is we have to be patient. We have to think about the long run of like
Right.
understand that discoveries now we probably won't see payoffs until the future. And
Sure.
so where the opportunity to improve this is to reduce what's called the translational gap. So,
Yeah.
working in my dissertation, we were focusing on a phytochemical compound that purportedly in cells and like very basic rodent models had potential to improve mitochondrial function.
Okay.
And so, usually, when you make a discovery like that, it takes about 17 years to move it to a translational to like from to bedside. So the bench to bedside gap, the translational gap is about 17 years.
Oh wow.
If there was better communication and better collaboration, I think we could reduce that a little bit better. And sometimes that means picking better model organisms
Mhm.
preclinically, while also thinking about parallel safety studies to make sure that it is safe to give in perhaps more complex organisms that resemble humans, so that when or if we see positive effects, we're in a better position to determine the safety of this drug or this intervention. When it comes to, say, more clinical outcomes, I think where academic institutions can really help is that we've got quite a workforce. Or,
Yeah.
we've got a lot of future healthcare workers, future scientists. When I was a graduate student, I helped pilot one of the CGMs for Dexcom, and it was just a collaborative opportunity. A postdoc in my lab and the two PIs that ran the lab had facilitated with that institution, and like that was a really exciting training opportunity for me. But they also got a lot of valuable data from that too, because we were doing exercise training interventions that they hadn't done before in adults, and so they could use that data to help inform their device and their algorithms. And
Sure.
so I think it's a matter of making sure we stay out of our silos and to try to see if we can find a solution elsewhere instead of just waiting for it to come to us necessarily. It was a very
No.
long-winded answer, but I distill it down, more collaboration and this accelerationist sort of like, let's try to get this to a patient or to an intervention sooner.
Sure, absolutely. No, I think you really do make it right because the bench to bedside model it's great, yeah. But again, it's yes, you have a concept, then you typically go into an animal model. That animal model can take a couple years. It especially has to go through that, and before it can even be introduced into a non-animal or non-primate human model, right? So it does take quite a bit of time, and
Yeah.
yeah, I would love to see that be expedited a little bit too. So. Great, but one thing as we kind of start to land this plane here, I'd really like to kind of end really on your academic side and says, you know, you teach a lot. You mentioned earlier, you're 50-50 teaching and research, but what advice would you give to students either entering the field of exercise physiology, human performance, or maybe some that are on the cusp of, maybe I don't know what I want to do just yet. I like working out, things like that. So what advice would you give to those students or aspiring professionals?
Yeah, I think it's sort of the same advice that I try to give my students, and that is to um, be open to what interests you the most. It's not the same thing as saying like follow your passion, because I think that's quite often said. Um, but find what motivates you and interests you. I thought for the longest time that I wanted to be a clinician,
Okay.
and I was driven by the fact that I wanted to help people.
Sure.
I didn't love the exposure to clinical experiences that I had, it felt very repetitive, and it just didn't feel like it resonated with me. What I enjoyed about research was the general novelty of everything we did and the challenge and the puzzle of fitting that together. And so I often shared my trajectory with students to demonstrate that you may not think you want to go and do this, or you may think you want to do something else, but be open to being persuaded otherwise. And I think that's a really important aspect. And then the other thing is, don't hesitate to really sink yourself into it and to really push yourself, and know and find some people, so find some mentors that are just going to support you as you struggle, because getting from like early 20s to mid 20s to 30s to a career that you enjoy is a lot of hard work. There's a lot of heartache. There's a lot of sleepless nights. Like there's a lot of stress involved in that.
Yep.
And so finding that network, that support is so critical.
No, absolutely. Now, finding that good mentor, hanging on to them, letting them, still being able to call, text, email, even after your tenure together, things like that. I really agree with you on that regard. So thanks for sharing that. So again, Dr. Musci, as we start to kind of close out here, where can people find you for collaboration? Do you have any social media channels you want to direct folks to, or websites, anything like that? We can also include it in the show notes too. But,
sure. Yeah. So I'm pretty bad on socials. I'm pretty bad on websites. I've got an email, robert.musci@lmu.edu. I have like a blank lab account, like lab website. It just hasn't been filled out yet. Eventually, I'll get there. But I'm always open to collaboration. That's like the best part about research. I think it was one of the things that excited me most when I was getting into this field was the opportunity to travel, to meet new people. I think despite sometimes thinking of myself as an introvert, I love the extroverted nature of science when we get to collaborate.
Sure. No, and again, the collaboration, moving the science forward, working with folks who might not necessarily be in your exact field, but have a link and can help, again, fill a gap or fill a space. That's really the best part, especially within the whole ACSM kind of network and the country, everything like that. It's just really an awesome part to be in. So, well, great. Well, Dr. Musci, again, thank you so much for your time here on the Electra-Med Edge. We look forward to seeing you at future meetings and really starting to track your research and publications as they come to publish.
Yeah, thanks for having me. I really, really appreciate it.
You're so welcome. Thanks again for tuning into the Electra-Med Edge, where innovation meets precision in human performance and clinical care. To learn more about Electra-Med's full range of medical equipment and supplies, spanning neurology, cardiology, physical medicine and rehabilitation, sleep diagnostics, and respiratory care, visit us at electramed.com. Also, check us out on LinkedIn and Instagram at simply @Electra-Med. Be sure to follow, subscribe, and share this episode to stay on the cutting edge of healthcare technology. Thanks again for joining us, and we'll see you on the next one.